Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today the video is related to microbiology, and the title of my today's lecture is the Indole Test and its mechanism. Indole test is basically a biochemical test that is used to detect species of Enterobacteriaceae in a specific inoculum. Okay, yeah, it is used to test an inoculum that which type of bacteria it is present, which type of bacteria is present in it. So if it is, if it is the member of Enterobacteriaceae family, so it can be detected through indole test. Okay, so in today's video, I will discuss that what is basically indole test, what is the principle, and how it is performed in the laboratory, and at the last I will show you the chemical reactions that is the most important part of this video, that what happens inside the indole test, what are the chemical reactions that are involved inside the indole test. And at the last I will give you the examples of different bacteria that give positive indole test and negative results of indole. Okay, so let's start with the video lecture. First of all, you should know about that what is basically an indole test. So this test basically demonstrate the ability of a certain bacteria to decompose the amino acid tryptophan to indole, which accumulates in the media. In simple words, it means that we detect a special type of enzymes that are present in specific bacteria, and mainly that enzyme is present in the members of the family Enterobacteriaceae. That enzyme is known as tryptophanase enzymes. Okay, and those enzymes are used to degrade tryptophan that we give, that is used in the indole media. Okay, so when we give tryptophan to the bacteria, so those bacteria will decompose it, will metabolize it if they contain tryptophanase enzyme. So after that, when it is degraded, so it is converted into indole, and after that, with different procedures, we can detect the in presence of indole. Okay, I will discuss it in the later video. So in simple terms, the principle is that we have to detect the tryptophanase enzyme, that either a bacteria consists of tryptophanase enzyme or not. If it consists of a tryptophanase enzyme, so it is a member of Enterobacteriaceae, and if it does not consist of tryptophanase enzyme, so it is not the member of Enterobacteriaceae. So it gives a negative indole test. Okay, so this is the principle of it. So indole production test is important in the identification of the member of Enterobacteriaceae. Most strains of the E. coli, Pseudomonas vulgaris, and Pseudomonas returgis, and other type of bacteria species break down the amino acid tryptophan with the release of indole, as I have told you. Okay, so these bacteria give positive indole test. So this is performed by a chain of number of different intracellular enzymes, a system generally referred to as tryptophanase. Okay, so as I have told you that basically we have to detect these enzymes, this enzyme complex that is known as tryptophanase. So we are conducting this test. Okay, so it is used as a part of the IMVIC procedure. Basically, this is a combination of biochemical tests. So I stand for indole, and M stand for methyl red, and V stand for Voges Proscus test, and the C stand for citrate test. Okay, so in I means indole. So indole test is a part of these biochemical reactions or biochemical test. Okay, so this is an informative text, and these tests are. If someone asks you that what is IMVIC procedures, so you have to tell that these are the combination of biochemical tests that are used to distinguish between the members of the species of the Enterobacteriaceae. Okay, so it is an MCQs and it can come in the paper. So now here come to the procedure. If you are working in the laboratory, so how you will start the indole test. Okay, if your examiner asks you that please perform an indole test, So what will you do? So there will be a bottle that will in which there will be powder type of substance, and basically on the upper part there will be written indole. Okay, so what will you do? Or indole powder or indole citrate powder, so different type of writing are there according to the company protocols. Okay, so you have to take that powder and you have to dissolve it in water. Okay, in distilled water you have to dissolve it. And one thing, keep this thing in mind, that it does not consist of agar. Okay, so indole, the media of the indole will never be solidified. Okay, it will be, it will never be solidified, it will be in liquid form. So what will you do? You will add the powder, and after that you will place the beaker in the incubator in order to kill all the bacteria and all the spores and all other type of microorganism that is present in that water or on the beaker. And you have also one thing, you should also in a incubate sorry, sterilize the test tubes. Okay, because you have to pour the media into the test tube. Just look here, this man is pouring the media into the test tube. So after the after the autoclaving, you have to pour the indole media, the indole broth media into the test tubes. It's up to you that you have to take two test tubes, three test tube, or four test tubes. It's up to you. It rely on the on the amount of the inoculum, that how much species you are going to detect it. And after that, when you add the indole media into the test tube, so now this is the color of the indole media. Okay, you have added it into the test tube. So what will you do? After that, you have to streak, you have to inoculate, or you have to streak your desirable or your unknown culture into that indole media. Okay, so what will you do? You have to take it on inoculating loop. Okay, a wire loop. You should take the media, sorry, the bacteria, the culture from the culture. And after that you have to dip the inoculating loop into the just look here, you have to dip it into the indole media. Okay, so now you are dipping it. So you have to dip it up till here, but keep this thing in mind that you should not touch the bottom of it. Okay, the bottom of the test tube. After that, you place it for 3 to 5 seconds in the test tube. Then you have to remove the inoculating loop, then place a head over that. If you are having a head, so you have to place it on the test tube. Or if it is headless, so you have to put a cotton on on the head of the on the surface of the test tube. So this was basically the when you are working in the laboratory. So you have to perform this such type of procedure. And then after that, you have to place the test tubes in an incubator. Okay, in order to grow that microorganism that you have inoculate it into the test tubes. This is the procedure of indole test in text form. Okay, so as I have told you in the previous slide, in the animated form, this is all about these three steps, that how you should perform an indole procedure. So you can use these slides in your assignments and your presentations. After that, now here come the main step. What will you do? That when after one day or after 20 hours, you have to take out the test tube from the incubator, and then you have to add a special type of reagent that is known as Kavox reagent to the broth culture. Okay, to the you have to add a few drops up to 0.5 milliliter. It is equal to 3 or 4 drops. Okay, so you have to add 3 or 4 drops into the test tube. Okay, that is the broth. Okay, the broth that is basically the indole. Observe the presence or the absence of the ring. Now within few seconds you will find the result. You will There will be a special appearance just like this. Okay, so if your bacteria is indole positive, so there will be an orange color on the surface of the test tube. And if your bacteria is indole negative, so there will be just a yellow color. There will be no orange color. Okay, so it is indole negative and it is indole positive. Okay, so here is up till this slide, it was just a technique. Now for a real microbiologist, you should know that what basically happens inside the test tubes, what basically the bacteria do with that coac region. Okay, and what is basically a coac region. So now we will come here to the chemical reactions. Okay, this is the main part of the video. So what will happen that you have in your indole media, there is plenty of tryptophan. Okay, you have given it to the bacteria, and there is also water. Okay, so if your bacteria consists of tryptophanase enzyme, okay, if it consists of tryptophanase enzyme, so it will metabolize and break down the tryptophan to indole, pyruvate, and ammonia. Okay, ammonium ion. It will break down the tryptophan into indole, pyruvate, and ammonia. If this enzyme is present, so this chemical reaction will proceed. Okay, so now here the indole is produced. So what will happen that when indole is produced, so after that when we take out the test tube from the incubator and we add coac region. Okay, we add coac region to the test tube. So now here is the 2 to 3 or 4 drops of coac region. So if we are having indole, okay, so what will it do? It will react with the indole. This coac region, basically it is dimethyl aminobenzaldehyde. It will react with the indole, and after chemical reaction there will be a complex that is known as quinidine red foilate complex, and it is basically red in color. And just because of this red color, this is just because of this complex that is formed just because of the binding of indole to the coac region. And then we can easily detect it that our bacteria is indole positive. Okay, so if there is indole, it will react with the coac region, and what will happen that an orange color will be produced. Okay, or if our bacteria is indole negative. Okay, if our bacteria is indole negative, so what will happen, there will be the bacteria will not be consisting of tryptophanase enzyme. So if there are no tryptophanase enzyme, there will be no indole. And if we add coac region to the if we add coac region, so it will not react with the indole because we are not having indole. So what will happen, there will be no color formation. Okay, even there will be a ring, but it will not be in red in color. Okay, and one thing I want to tell you guys that why this ring forms, basically the coac region consists of a amyl alcohol, and amyl alcohol is a hydrophobic. Okay, it does not dissolve in the water, and it is present on the surface of the water because it is not dissolving. So what will happen that the coac region will be present on the upper surface of the end. If it is so, the coac region consists of amyl alcohol, so it will be present and it is hydrophobic. So what will happen, so it will not react with the water, and it will remain on the upper surface of the water. So if there is indole, so it will react with the coac region and the complex will form. And if there is no indole, so it will be present, or the coac region will float on the upper surface of the water, but it will not react with the indole because there is no indole and no chemical reaction will proceed. Okay, so keep this thing in mind. If someone asks you that why the coac region is floating over the surface of the water, so you will your answer will be that it consists of amyl alcohol that is hydrophobic and it cannot be dissolved in the water. Okay, just like oil. So that's why. So it was the chemical reactions, and these are the positive. This is the text form. If there is a cherry red like ring, so the it is indole positive. And these are the example of the bacteria that are indole positive: aromonas hydrophila, pancata, bacillus lv, elvera, e coli. Okay, hemophilus influenza, Klebsiella, these all are indole positive, and these bacteria are indole negative. Okay, just look here. If no color change even after the addition of the appropriate region, that is coax, and if there is no color, so it is indole negative. So these are the examples of the bacteria that are indole negative. So you can use these slides in your assignments and your presentations. Okay, so this was all about the indole test. We have discussed the principle of it, the procedure of it, and I have also added the text form. And at the last, we have the most important part, that we understand the chemical reaction that is taking place inside the Indole test, and why red colors appear and why there is no color. So we also discuss it there. Okay, so if you are having any further questions, so you can ask me in the comment box. And if you have understood this video, so like this video, share it with your friends, and subscribe to my channel for more interesting videos related to science. Thank you very much. God bless you.